Hello, everyone. My name is Paige Ballack. I manage what's called the Keck Bioimaging facility. It was the Keck Lab in the School of Life Sciences, and we've just recently changed that to the Regenerative Medicine Imaging Facility. There are two microscopy cores, and so mine is the Regenerative Medicine Imaging Facility. The general name of the whole core of microscopy is the Advanced Light Microscopy Core. And within our counter would be in biodesign, and that one is managed by Honor Glenn. And so it's the biodesign imaging facility. And so within the regenerative medicine facility, we have a large range of microscope systems, including things such as the fluorescent wide field, confocal, multiphoton, live cell, and light sheet microscopy. Over in biodesign, we have some additional modalities, which include now a new Zeiss 880 ARI scan. They also have structured illumination, TURF systems, which are total internal reflection microscopy, and FLIM now, and that's fluorescent lifetime microscopy. So we have a lot of tools that you could use if you're interested. Another thing I wanted to point out is in the little chat area, I put the email address for the microscopy course. He develops in vitro molecular assays as part of functional validation team uncovering genetic etiologies of cerebral palsy. Throughout his presentation, Dr. Guida will highlight the impact that access to multimodal core facility resources has benefited his interdisciplinary research. We wish to welcome Dr. Brandon Guida as he presents.
So as Paige mentioned, I'm going to be really talking about how access to all of the different instrumentation at ASU has impacted my ability to be an interdisciplinary. I've worked on 3 different projects, and what I'm really going to do is just give a broad overview and show some of the images that I've collected when I was doing some various experiments here. We definitely don't have the time, and I definitely don't want to put any of you to sleep by talking too much in depth about this info. So going and starting at the infectious disease side here in Shelley Haydell's lab, we were really looking, we were studying how the deletion of certain 2-component regulatory systems. Now, these are genetic systems which produce proteins that are sensors. And these types of proteins are ubiquitous in microbiology. Almost every microorganism has multiple sets of these sensors. And these sensors are relaying environmental information from the outside to the inside of the bacterium. And then the bacterium is using that information to modulate gene expression in order to adapt to changes in the environment. So then, because of the different imaging instrumentation that was available at ASU, I was able to try to look using a different type of microscopy, and in this case, it was scanning electron microscopy. So with the help of, with David Lowry, who runs one of the electron microscopy cores, we were able to come up with a way to actually take a scanning electron microscope image of an entire colony of tuberculosis as it grows on the Petri dish, right? And so this image on the left is actually one of those pictures, and all the way into the top left, you can actually see this is the agar plate kind of in the background. And we know that TB, as well as a lot of other microorganisms, they shed extracellular vesicles, and those are color-coded in pink here. We don't necessarily know the function of these extracellular vesicles in the context of growing on an agar plate, but we do know they have something to do with pathogenicity when in the body. So really, what I was trying to do is take over his work and just kind of continue what he had discovered with this particular organism. Now, this organism is a U endolith, right? U means true, endo means inside, lith means rock. So this is a microorganism which can bore, actively tunnel its way into certain types of rocks. And in this case, this organism, it's calcium carbonates. So it's dolomites, aragonites, things like that, calcite, and a lot of this, limestone, and there's a lot of this material in a lot of the oceanic shelves in the Caribbean, for instance. So I got to spend a lot of my time on a Caribbean island for my dissertation. It was horrible, absolutely horrible. But we investigated the mechanisms and we tried to figure out how exactly does this microbe bore into the rock. And we used a lot of different techniques for this, but we really wanted to look at how calcium was moved in and out of the cells. And we wanted to look at how the pH changed when the calcium moved. We also really wanted to see if we could see where within the cell the pumps might be for calcium. And we'll talk about that in a minute. And then the other thing we really wanted to see was, why in the world would this microbe want? And we then kind of came up with the hypothesis that, well, these rocks are made out of a lot of carbon. Maybe they're actually using the carbon and they're eating it. So remember, these are phototrophic organisms. So they fix carbon dioxide and they turn that into sugars, which then they use for their own biology. And confocal was amenable because this particular organism bored down into the calcium carbonate, but it could only bore as far as light could penetrate. So it was really nice to be able to use confocal to be able to look at how the organism lived within the crystal. And on the right-hand side here, you can see what I did is I used one of the channels in the confocal to collect laser light that was reflected back to me from the chip so that I could get a topological view of the calcite here. And so in the gray is the actual rock itself, and then the red is the autofluorescence from the chlorophyll. Another thing we really wanted to do was we wanted to see where in the organism these calcium channels might be. So previously Ed had showed that this organism could move calcium between the two ends, so the boring front and then the front that's outside sticking into the medium. And we wanted to see, could we actually find where those channels are? And when we incubated our cells with Bodepi topsy-garden, we found that there were some tips, and we call these apical tips, that lit up like a Christmas tree. They had a very high concentration of these P-type calcium ATPases. Not every tip, right? But some of them. But we had a dipping objective. And so this was something that we could put on the microscope, and we could actually put the lens in the liquid medium, in the seawater that this organism lived in. And this allowed us to look at how calcium was being moved in the cell in real time, in situ, in a living organism. Fueled by the fact that ASU has so many experts and so many cores, I was able to start asking questions of people. Okay, how can we figure this out? How can I interrogate the way that this organism uses carbon? And the best thing we could do was use isotope tracing. So we used C13, which is a slightly heavier isotope of carbon, and we looked at how carbon was transported when we grew the organism in a C13-rich environment. And basically, what we found using isotope ratio mass spectroscopy, and we also used nanosims. The nanosims is a highly sensitive instrument which combines transmission electron microscopy with the ability to actually look at the ratio or the numbers of different isotopes of different. These are nuclei of glial cells. So these are supportive cells for the neurons. And what we can do is we take our, I'm going to call it gene 1 because this is not published data yet. So gene 1 here, we took it and we put it into the same cells and we overexpressed the normal form and we overexpressed the mutated form. And all we're trying to do here is see if the mutated form does something different than the wild type form. Okay? That's all we want to, that's all we care about at this point. And indeed, if we take these images and we do something to them to help us analyze, right? We take the neurons out and we do something called skeletonization. So we can take the neurons out, and so now we can just see the neurons without all the background. And if you look at them, you can definitely see that the mutant overexpressing cells look much different. So this is wild type fibroblasts here. And we can coax them to turn into fat cells. And we can then label the fat with a dye. In this case, the dye is kind of a red-orange color. Right? The redder or the brighter the orange of the cell here, the more fat that's inside of the cell. So here we've got our wild type, no manipulation, no overexpression of anything, just normal fibroblasts. And then here we can knock down the gene we're interested in, and we see there is a difference in the fat accumulation during adipogenesis. And then this is the patient fibroblast line, right? So you just take a punch biopsy from a patient, you take it out, you chop it up, and then you can grow fibroblasts from it. And we notice that when we quantify, right, all of the oil red O that we see in these cells, and we plot that, we can see that, well, the negative controls are all the same. If we knock down the gene, we have a lot less fat. But in the patient cells harboring this mutation in this gene, we have a lot more fat accumulation, right? So these were taken on the Leica SP8. These are 3 individual images, and then you can stitch them together from the same well, and then you can do your quantification. Thank you very much for curing your boredom with me today in this tech talk, and then we'll open it up for any questions that anybody has.